Now we have chosen to study the module on the production of fatty acids and amino acids. Traditional or old biotechnology has been employed for the preparation of foods and beverages and were based on the natural capabilities of the microorganisms, even though their existence was unknown at that time. Many of the products from the microorganisms have multiple functions in the human life. Fatty acids and amino acids are the important products that have a great benefit to the mankind. Amino acids are consumed in a variety of markets. The largest by volume is the food flavoring industry. The basis of our ability to enjoy the taste of amino acids is rooted in evolution. Therefore, after completing this module, you will be able to understand the production of fatty acids. You will be able to comprehend the various types of amino acids produced by the microbes. You will understand the applications of fatty acids and amino acids in the food industry. To start with fatty acids that occur in food are straight chain monocarboxylic acids with an even number of carbon atoms. The acids are either saturated or unsaturated with 1, 2 or even up to 6 double bonds. The short chain fatty acids occur as triglyceride constituents in milk fat, coconuts and palm seeds. They are used by the food and beverage and pharmaceutical industries and can also be converted chemically or enzymatically into important biofuels, including fatty acid methyl esters, fatty alcohols, methyl ketones, alkenes and alkanes. Research on the bio-based production of fatty acids has been mostly conducted in E. coli and Clostridium. The three main approaches that have been used are diversion of phospholipid biosynthesis, reverse beta oxidation and fermentation. Coming to the production of fatty acids, they have been produced by microbial fermentation. Certain bacteria have native fermentation pathways that produce butyrate and hexonate and have been used for the production of these fatty acids. Clostridium tyrobutyricum was used to produce high levels of butyric acid from various substrates in a fibrous bed reactor. Clostridium species and Megasphaera elsdeni were used to produce hexanoic acid from D-galactitol and sucrose respectively. In addition, Clostridium acetobutylicum has been engineered for high-level butyrate production. However, the development of Clostridia for efficient production of diverse fatty acids is encumbered by slow growth, a requirement for strictly anaerobic culture conditions, sporulation and relatively limited genetic tools. Consequently, butyrate production was engineered into E. coli. The reaction sequence used was similar to the butyrate fermentation of Clostridium to the point of butyryl-CoA. Next, let us see how the metabolic engineering of microbes occur. Metabolic engineering of microorganisms has provided a platform for effective production of these valuable biochemicals. Notably, synthetic biology-based metabolic engineering strategies have been extensively applied to refactor microorganisms for improved biochemical production. There are two strategies. One, the current status of metabolic engineering of microbes that produce fatty acid derived valuable chemicals, and second, the recent progress of synthetic biology approaches that assist metabolic engineering such as mRNA secondary structure engineering, sensor regulator system, regulatable expression system, ultra-sensitive input-output control system and computer science-based design of complex gene circuits. Fatty acids are one of the major components found in all organisms, usually in intracellular forms of fatty acyl carrier protein called as acyl ACP, fatty acyl coenzyme A ester, acyl-CoA, storage lipids, eicosanoids and unesterified free fatty acids. In industry, applications of free fatty acids are generally limited due to the ionic nature of their carboxyl group. The production of fatty acid derived chemicals by engineering microbial cells into microbial factories is becoming an attractive alternative approach that can overcome the aforementioned bottlenecks associated with the other synthesis routes. To date, synthetic enzymatic pathways that led to the production of fatty acid derived valuable chemicals including fatty alkanes, fatty acid methyl ethyl esters, fatty alcohols, hydroxy fatty acids and lactones have been constructed in microorganisms such as E. coli and Saccharomyces cerevisiae. Omega-3 fatty acids, they are known as essential fatty acids because they are important for good health. They have many positive effects on human beings such as anti-inflammatory and anti-blood clotting actions, lowering triglyceride level, reducing the blood pressure and also reducing the risks of diabetes and some cancers too. The human body cannot synthesize these fatty acids on its own. Therefore, the omega-3 fatty acids must be obtained from the diet, that is eicosapentaenoic acid and docosahexaenoic acid or two typical omega-3 fatty acids. Their traditional source is derived from cold water fish oils. Apart from the algal or microbial fermentation procedure, the omega-3 biotechnological process also includes efficient and green downstream procedures, that is oil extraction and refining process. The oil product rich in omega-3 fatty acid obtained is a kind of intracellular metabolite. Therefore, before extracting the oil from the algal biomass, the cells must be disrupted first. Traditionally, the method used was mechanical based and its high energy requirements pose a major challenge. Now, let us move on to amino acids. Amino acids, they are the basic bioelements of proteins, which are the most important macromolecules for human and animal functions. The production of amino acids is carried out by any of the following three processes. First one, the extraction. Amino acids are the building blocks in protein structure. The proteins can be subjected to hydrolysis and the requisite amino acids can be isolated. Example, cysteine, tyrosine and lysine. And second is the direct chemical synthesis method. Chemical synthesis results in a mixture of D and L amino acids. Most of the amino acids required for commercial applications are of L category. However, for the synthesis of glycine, that's optically inactive, and some other amino acids which can be used in L or D form, that's called as DL-alanine and DL-methionine, for certain purposes chemical methods are employed. Let's see the fermentative production. For the large scale production of amino acids, microbiological methods are employed. And here again there are three different approaches. The first one, direct fermentation methods. Amino acids can be produced by microorganisms by utilizing several carbon sources. Example, glucose, fructose, alkanes, ethanol, glycerol and propionate. Certain industrial byproducts like molasses and starch hydrolyzed can also be used. Methanol, being a cheap carbon source, is tried for amino acid production but with limited success. And second approach is the conversion of metabolic intermediates into the amino acids. In this approach, the microorganisms are used to carry out selective reactions for amino acid production. Example, conversion of glycine to serine. And the third approach is the direct use of microbial enzymes or immobilized cells. Sometimes resting cells, immobilized cells, crude cell extracts or enzyme membrane reactors can be used for the production of amino acids. Some examples are given below. Amino acid dehydrogenases from certain bacteria, example Bacillus megaterium, can be used for the amination of alpha keto acids to produce L amino acids. Example, alanine from pyruvate, phenylalanine from phenylpyruvate. Immobilized cells or enzyme membrane reactors can be used. Enzymes or immobilized cells are also employed for the production of several other amino acids. Example, tryptophan, tyrosine, lysine and valine. Next, let us discuss the development of microbial strains for amino acid production. The metabolic pathways in the microbes for the synthesis of amino acids are controlled in a systematic manner. Therefore, the organisms as such produces only less amount of the amino acids. Some strains that excrete certain amino acids have been isolated. Example, glutamic acid, alanine and valine. Hence, the microbial strains can be modified to produce the amino acids in a higher quantity by several methods. Many amino acid producing microorganisms have been developed by mutagenesis and screening programs. The following are the major ways of strain development. So, to develop a new strain for producing amino acids, a combination of methods can also be adopted. So, the first method is isolation of auxotropic mutants. These mutants will have the metabolic pathways where the repressor will not be produced, due to which the feedback control mechanisms are completely removed. The intermediates of the metabolic pathways, they accumulate and get excreted. And the second method is the isolation of recombinants. Mutants can be developed by genetic recombination for overproduction of amino acids. Protoplast fusion in certain bacteria is used for development of hybrids. Example, Corynebacterium glutamicum and Bacillus flavum. Now, we shall look into the amino acid glutamic acid. Glutamate is the most abundant free amino acid in bacterial cytoplasm. Glutamate producers must overproduce glutamate in excess of their normal metabolic needs, and they must excrete it into culture broth. Members of the actinobacteria, the genus Corynebacterium, were the most effective producers. Over the years, various glutamate producing bacteria have been isolated and classified as Orthobacter, Brevibacterium, or as members of other genera, but recent work has shown that almost all of these strains belong to the genus Corynebacterium. Wild-type cultures produced up to 10 grams per liter of glutamic acid. Yields were quickly improved by process engineering and by developing overproducing mutants. Yields are now in excess of 100 grams per liter. All these organisms have certain morphological and physiological characters comparable to Corynebacterium glutamicum, and biochemically, glutamic acid producing bacteria have a high activity of glutamate dehydrogenase and a low activity of alpha-ketoglutarate dehydrogenase. They also require the vitamin biotin. Several improvements have been made, particularly in Corynebacterium glutamicum, for improving the strains to produce and excrete more and more of glutamic acid. These include the strains that can tolerate high concentrations of biotin and lysozyme sensitive mutants with high yield. Let us see the production of L-glutamic acid. The pathway for the synthesis of glutamic acid with glucose as the carbon source is depicted in figure. Glucose is broken down to phosphoenol pyruvate and then to pyruvate. Pyruvate is converted to acetyl-CoA. Phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxylase can be independently converted to oxaloacetate. Both these carboxylation reactions are quite critical and require biotin as the cofactor. In Krebs cycle, the key metabolite, namely alpha-ketoglutarate, is produced, which is acted upon by the enzyme alpha-ketoglutarate dehydrogenase to form succinyl-CoA. For the production of glutamic acid, alpha-ketoglutarate is converted to L-glutamic acid by the enzyme glutamate dehydrogenase. Actually, one molecule of glutamic acid should be formed from one molecule of glucose, but the conversion efficiency of glucose to glutamic acid was found to be only 70%. Let us see the regulation of glutamic acid biosynthesis. The essential requirement for glutamic acid production is the high capability for the supply of the citric acid cycle metabolites. This is made possible by an efficient conversion of phosphoenol pyruvate as well as pyruvate to oxaloacetate. Thus, there are two enzymes, phosphoenol pyruvate carboxylase and pyruvate carboxylase, to efficiently produce oxaloacetate, while there is only one enzyme, pyruvate dehydrogenase, for the formation of acetyl-CoA. Certain microorganisms which have either phosphoenol pyruvate carboxylase or pyruvate carboxylase, they are not capable of producing glutamic acid to any significant extent. Corynebacterium glutamicum has both the enzymes and therefore can replenish citric acid cycle intermediates through oxaloacetate while the synthesis of glutamic acid occurs. Another key enzyme that can facilitate the optimal production of glutamic acid is alpha-ketoglutarate dehydrogenase of citric acid cycle. Its activity has to be substantially low for good synthesis of glutamic acid, as is the case in Corynebacterium glutamicum. Further, exposing the cells to antibiotics and surfactants reduces the activity of alpha-ketoglutarate dehydrogenase, while glutamate dehydrogenase activity remains unaltered. By this way, oxidation of alpha-ketoglutarate via citric acid cycle can be minimized, while the formation of glutamic acid is made maximum possible. Glutamic acid is synthesized intracellularly and therefore its release or export is equally important. For many years, increased membrane permeability was thought to promote glutamate excretion. Recently, however, export via a specific efflux transporter has been proposed as a mechanism for avoiding exceptionally high levels of intracellular glutamate. Such an exporter has been proposed by Analogue with the lysine E transporter that is responsible for lysine and arginine export in Corynebacterium glutamicum. Other factors contributing to glutamate overproduction include metabolic flux alterations based on cell growth limitations. A triggering mechanism like biotin depletion, high temperature, cell membrane alterations by detergents, oleic acid or antibiotics results in a decrease or repression of alpha-ketoglutarate dehydrogenase. The result is a redistribution of metabolites at the branch point in the TCA cycle, leading from alpha-ketoglutarate to succinyl-CoA or glutamate. The increase in glutamate levels beyond that which is needed for cell growth, it stimulates glutamate efflux as the cell attempts to maintain the proper level of intracellular glutamate. The result is the excretion of glutamate from the cell. Let us consider the process of production and recovery. The process is most commonly run as a fed batch type where sugar is added during the fermentation process. The reason for using fed batch rather than batch, where all components are present at the beginning of the process, is that high sugar concentrations, that's more than 20% in total, would otherwise need to be added to the medium. Such high levels can lead to incomplete oxidation of the sugar to lactic or acetic acids or to osmotic inhibition of growth with subsequent decrease in yields. Small seed cultures are grown on glucose, potassium phosphate, magnesium sulfate, yeast extract and urea as a source of nitrogen. Larger cultures use cheaper sources of sugar including cane or beet molasses and starch hydrolysates from corn or cassava. The sugar source roughly parallels the geographic location of the process. Corn syrup, tapioca and cane and beet molasses can also be used. Ammonia and ammonium sulfate are also used as nitrogen sources. Ammonia also used to control the pH during the fermentation. Cheap sources of vitamins and other nutrients include cornstrip liquor, a byproduct of cornstrip manufacture that is supplied with amino acids, nucleic acids, minerals and vitamins. To begin the process, stalks of Corynebacterium glutamicum are used to inoculate the shake flask cultures. The resulting cells are transferred to a small seed culture tank that's grown and used to inoculate a larger tank, and so on. The intermediate seed culture volumes are variable, generally in the range of 200 to 1000 litre, then 10,000 to 20,000 litres, and finally 50,000 to 5 lakh litres. The process is carefully controlled at each step such that cell density, nutrient composition, temperature, pH, aeration, agitation rates, and sugar flow rates are as consistent as possible from batch to batch. Oleic acid might be added at the beginning of the fermentation to encourage glutamate excretion, and pH is 8.5 with ammonia and maintained at 7.8 during the process. After 14 hours of growth, the temperature is increased from 33 degrees centigrade to 38 degrees centigrade. Sugar is fed in as the fermentation proceeds up to about 36 hours. During the course of the fed batch process, about 160 gram or more of glucose per liter or its equivalent is fed into the bioreactor. Glutamate concentrations are monitored at intervals, and after the process is complete, the broth is pumped from the bioreactor to recovery tanks. A schematic representation of glutamic acid production plant is shown in the figure. As the fermentation is complete, the cells are separated, the culture broth is passed through an anion exchanger. The glutamic acid bound to the resins is eluted in NaOH, while the ammonia released can be reused. With NaOH, glutamic acid forms monosodium glutamate, which can be purified by passing through anion exchanger. Monosodium glutamate can be subjected to evaporation and crystallization. We shall discuss the factors influencing the production. The production of glutamic acid is influenced by carbon sources, nitrogen sources, growth factors, pH, and as well as oxygen supply. Next, let us look into the lysine. It is the second produced amino acid in a large industrial scale with several 100,000 tons of production per year. Since animal feed such as grain and defatted oil seeds contain only a small quantity of lysine, poultry, cattle, and other livestock are unable to synthesize this amino acid. As a result, it must be added to this feedstuff to provide an adequate diet. Moreover, it is used to enrich human foods which lack this essential amino acid. It is the first limiting amino acid in virtually every cereal grain known to man. Thus, it has large potential for improving the protein quality of cereal-based human diets, especially in third world countries, owing to their high dependence on cereal foods. Supplementation of wheat-based foods with L-lysine improves their protein quality, resulting in improved growth and tissue synthesis. In addition, it has pharmaceutical applications both in the formulation of diets with a balanced amino acid composition and in the infusion of amino acids. In the early 1960s, it was found that Corynebacterium glutamicum homoserine auxotropes produced lysine, thus providing the first viable fermentation process for lysine production. Today, these bacteria produce well over 10 lakh metric tons of monosodium glutamate and 6 lakh metric tons of lysine annually. L-lysine is predominantly produced by Corynebacterium glutamicum and to some extent by Brevibacterium flavum or Brevibacterium lactofermentum. Next comes the production of L-lysine. The pathway for the synthesis of lysine is complex and is shown in the figure. This metabolic pathway is involved in the formation of three other amino acids, namely methionine, threonine, and isoleucine. To understand why lysine is overproduced from a physiological standpoint, one must first understand the regulatory circuits that govern the lysine biosynthesis. The approaches or paradigms of rational strain improvement as contrasted with approaches that rely on random mutagenesis and screening. The pathway leading to lysine, also threonine, isoleucine, and methionine biosynthesis is initiated with the conversion of aspartate to aspartyl phosphate via the enzyme aspartokinase. The phosphorylated aspartate is then converted to aspartyl semialdehyde, which can be converted to homoserine by homoserine dehydrogenase or to diaminopimelic acid by a series of 5 enzymatic conversions and hence to lysine. Homoserine is the starting point for making threonine and isoleucine as well as methionine. The major control points for the metabolic flux to individual amino acid occurs at the level of aspartokinase and homoserine dehydrogenase. Now, we will discuss about the feedback inhibition and repression. Aspartokinase is feedback inhibited by the accumulation of threonine plus lysine. Homoserine dehydrogenase is feedback inhibited by threonine and its synthesis is repressed by methionine. Homoserine dehydrogenase mutants, they are homoserine auxotrophs that effectively lack the ability to make threonine, methionine and isoleucine, thus eliminating the feedback inhibition of aspartokinase by threonine. Analog resistant mutants resist the feedback inhibition effects of allosteric effectors. In the case of aspartokinase, mutants were derived that resisted analogues of lysine and also threonine. Other types of auxotrophs, for example, leucine and alanine, are also overproduced by the lysine, since their biosynthetic pathways compete for pyruvate. In 1996, the gene encoding a membrane protein which are involved in the active export of lysine, that is Lys-E, was cloned and sequenced. It is responsible for exporting lysine up a concentration gradient, eventually leading to the accumulation of as much as 1 mole per liter of lysine. Mutations in this gene cause lysine to accumulate in the cytoplasm but not in the extracellular medium. Introducing the gene into your multi-copy plasmid in Corynebacterium glutamicum leads to increase in the rate of efflux of intracellular lysine. Coming to the lysine fermentation, the industrial production of lysine is carried out in a manner similar to that of glutamate production. Sugarcane or beet molasses is used as a substrate for production. If the biotin level is low in the starting material, it must be added to a level of about 30 microgram per liter. If an auxotroph for any amino acid is used, that amino acid must be present so that the cells may grow but should not be present at high enough concentration to exert its inhibitory effects, such as feedback inhibition of the pathways used for production. Under optimal fermentation conditions, the yield of lysine is 40 to 50 gram per 100 gram of the carbon source. Aspartate semialdehyde, they have two fates: the biosynthesis of lysine and formation of 3 other amino acids, namely methionine, threonine and isoleucine. When homoserine dehydrogenase acts on aspartate semialdehyde, it is diverted for the synthesis of 3 amino acids. The enzyme dihydrodipicolinate synthase converts aspartate semialdehyde to piperidine 2,6-dicarboxylate. There are 2 distinct enzymes, succinylase variant and dehydrogenase variant, that can convert piperidine 2,6-dicarboxylate to DL-diaminopimylate, which later forms L-lysine. Coming to the regulation of lysine biosynthesis, there are certain regulatory processes in the production of lysine. First important enzyme is the aspartate kinase. This enzyme is controlled by feedback inhibition of the end products. 3 isoenzymes of aspartate kinase have been identified, one repressed by L-methionine, the second by L-threonine and L-isoleucine, and the third one being inhibited and repressed by L-lysine. The amino acid sequence and structure of aspartate kinase have been elucidated, and by genetic manipulations, it has been possible to create mutants that are insensitive to feedback regulation by L-lysine. Second important regulatory enzyme is dihydrodipicolinate synthase. This enzyme competes with homoserine dehydrogenase to act on aspartate semialdehyde. Overexpression of dihydropicolinate synthase has been shown to increase the production of L-lysine. Succinylase and dehydrogenase variants, the conversion of piperidine 2,6-dicarboxylate to DL-diaminopimylate is carried out by these 2 enzymes. At the start of the fermentation, dehydrogenase variant predominantly acts, and later succinylate variant comes into picture for the biosynthesis of lysine. Coming to the release of lysine, the export of lysine from the cells into the surrounding medium occurs through a lysine export carrier protein. There are different recovery processes for lysine depending on its application. An alkaline solution containing about 50% lysine can be obtained after biomass separation, evaporation and filtration. And second, a crystalline preparation with 98 to 99% L-lysine can be obtained by subjecting the culture broth to ion exchange chromatography, evaporation and crystallization. Typical strains today, they provide only conversion efficiencies up to 50%. Next, let's move on to L-threonine. Threonine is manufactured industrially by employing either E. coli or Corynebacterium glutamicum. With the mutant strains of E. coli, the product yield is better. Biosynthesis of threonine is depicted in the figure. Some of the reactions of this pathway are common for the biosynthesis of lysine and methionine besides isoleucine. Starting with aspartic acid, in a sequence of 5 steps, threonine is produced. Coming to the regulation of threonine, the regulatory reactions in E. coli for L-threonine biosynthesis have been elucidated. 3 isoenzymes of aspartate kinase separately inhibited by the end products have been identified. One by L-threonine and one by L-methionine and the next one by lysine. Further, 2 isoenzymes of homoserine dehydrogenase. One inhibited by L-threonine, the other inhibited by L-methionine, they are also known. A gene THR-ABC that encodes 3 polypeptides. One polypeptide possesses the activity of kinase and homoserine dehydrogenase, and the second homoserine kinase and the third polypeptide threonine synthase in E. coli has been identified. How to improve the strain is the efficiency of the producer strains can be increased by creating E. coli mutants with high level expression of this gene, that is threonine ABC. Further, mutants with minimal production of L-isoleucine also result in high yields of L-threonine. Let us discuss the production process of L-threonine. The culture medium containing glucose or sucrose, yeast extract and ammonium salts is adequate for L-threonine production. The sugar feeding has to be continued for good yield at about 60% of the carbon source. The downstream processing for the isolation of L-threonine consists of coagulation of the cell mass, filtration, concentration by evaporation and then crystallization. Next is the L-phenylalanine. Both E. coli and Corynebacterium glutamicum can be used for the production of L-phenylalanine. The biosynthetic pathway is quite complex and shown in the figure. An interesting feature is that the same pathway is responsible for the synthesis of all the 3 aromatic amino acids, namely tyrosine and tryptophan, besides phenylalanine. The synthetic pathway commences with the condensation of erythrose 4-phosphate with phosphoenol pyruvate to form deoxyarabinoheptylosonate phosphate. This DAHP in the next series of reactions is converted to chorismate, which can form L-tryptophan. Chorismate mutase converts this chorismate to prephenate, which forms L-phenylalanine through the participation of prephenate dehydrogenase. Prephenate also serves as a precursor for the synthesis of tyrosine 2. The genes responsible for the formation of the regulatory enzymes of phenylalanine have been identified. By employing genetic manipulations, strains for improved production of phenylalanine have been developed. Coming to tryptophan, there are different ways of synthesizing L-tryptophan, either chemical, enzymatic and fermentation methods. At present, large-scale manufacture of tryptophan is carried out by using the enzyme tryptophan synthase of E. coli. Tryptophan synthase combines indole with L-serine to form tryptophan. Indole is available from petrochemical industries, while L-serine can be recovered from molasses during sugar refinement. Mutant strains of E. coli with high activity of tryptophan synthase have been developed for large-scale manufacture of tryptophan. Direct fermentation process for tryptophan is produced by the fermentation employing Corynebacterium glutamicum or E. coli. The pathway is shown in the figure. Mutant strains of both these organisms have been developed for increased yield of tryptophan. The mutant strains for the overproduction of L-tryptophan can be developed. The production of tryptophan by Corynebacterium glutamicum was increased by introducing a second gene encoding anthranilate synthase, a key enzyme in its biosynthesis. The result is that the pathway becomes insensitive to feedback inhibition by the end products, leading to an overproduction of L-tryptophan. Coming to the aspartic acid, there is a growing demand for aspartate, which is a component of aspartame, a sweetener, besides its use as a food additive and in pharmaceutical preparations. The preferred method for aspartate production is enzymatic in nature. The enzyme aspartase converts fumarate and ammonia to aspartate. Although this reaction is reversible, aspartate formation is favored. The aspartase of E. coli is used. This enzyme is quite unstable. Immobilization of this enzyme in polyacrylamide or carrageenan enhances the stability of the enzyme and is used commonly. Immobilized E. coli cells with good activity of aspartase are also used for aspartate production. To conclude, this module has presented the importance of fatty acids and amino acids in food. Production of fatty acids and the various types of amino acid production has also been presented. This module has given a clear picture about the fatty acid and amino acid production.